Hello, everybody. In this lecture, we're going to discuss respiration. So let's get right into it. Now, we've got quite a few objectives, but by the end of this module, you should be able to define cellular respiration, describe what occurs in glycolysis, the Krebs cycle, and describe chemiosmosis and how it drives ATP production. List the similarities and differences between aerobic and anaerobic respiration, and compare and contrast aerobic and anaerobic electron transport chains. All right. So, generally speaking, cells get their energy through carbohydrates. And they do that through either cellular respiration or through fermentation. And here are some basic outlines where in cellular respiration, you have glycolysis, an intermediate step, Krebs cycle, and electron transport chain. In glycolysis, you have, or in fermentation, you have glycolysis and fermentation. Now notice these arrows, they show kind of how each of these processes feed into themselves and how things go back and forth. I don't expect you to understand all that right this moment, but it is a good starting place to kind of think about what's going on. And so in cellular respiration, which we're going to focus on here, it's going to have these four primary pathways. We have glycolysis, the intermediate step, the Krebs cycle, and the electron transport chain. And these processes happen in a couple of different places. For example, glycolysis is going to happen in the cytoplasm of the eukaryote and the cytoplasm of the prokaryote. While when we're talking about the intermediate step, Krebs cycle, and electron transport chain, they're going to happen in the mitochondria, right? And for bacteria, the intermediate step and the Krebs cycle happen in the cytoplasm. The electron transport chain happens in the membrane. Now, this is kind of cool because this right here is another link to the endosymbiotic theory, right? So we've got the electron transport chain in the cell membrane of the prokaryote, and we have it in the inner membrane of the mitochondria. We've got intermediate step and Krebs cycle in the cytoplasm, and we've got the intermediate step and the Krebs cycle in the matrix of the mitochondria. All right, so when we're talking about glycolysis, this is going to be the conversion of glucose to pyruvate and the generation of ATP, right? And so when we do this, there's an initial investment of 2 ATP. So this is that activation energy that you need to get the actual process started. But by getting that glucose broken down to begin with, we can eventually get over to these 2-pyruvate and getting some more ATP. So this is what the pathway looks like. There are 10 reactions. There are 2 basic stages. There's the energy investment stage and the payoff stage. So the energy investment stage is from here up, and then the payoff stage is from here down. And then we're going to go over these a little bit closely, but you can see investment. This is ATP to ADP. This is that initial investment of energy to basically convert this glucose to glucose 6-phosphate. And then we have another investment of energy here where we do this fructose 6-phosphate to fructose 1,6 bisphosphate. So they get these 2 phosphates. And then from here down, we get our energy production. We get the 2 ATP generated here and 2 ATP generated here. And so this is just kind of showing you that a little bit closer up. During that energy investment phase, we do use 2 ATP molecules. So at this point, we have lost energy and we have not gained energy. And so if you think back to that, activation energy, exergonic and endergonic reactions, this is the part that you get over the hump, right? So you need this to get up to the hump. Now here, we're going to gain energy, right? So we've got 4 ATP produced. So we got 2 here and 2 here. That means that you get 2 net ATP from glycolysis because we lost 2 ATP in the energy investment stage. And so this is the bottom part of that graph. So as that ball rolls down, it actually rolls down further on that energy investment chart than it started at. And that's where you get this additional ATP. Now, also in this process, we do get NADP or NAD plus to NADH. So we get this reduction reaction where NAD plus is converted to NADH. And in doing that, we lose those electron carriers, and they'll need to be recharged later on. And as a quick summary, this is the important part here is that your glucose is going to go from a 6-carbon sugar to 2 3-carbon sugars that are pyruvates. It's going to produce 2 ATP, and your NAD plus is going to become NADH. It's going to be reduced NADH. So then there's this intermediate step. So we've got pyruvate. We've actually got 2 pyruvate. And the pyruvate is going to need to be converted to acetyl-CoA. Acetyl-CoA can enter into the Krebs cycle. It also produces CO2, right? And it does this again by reducing NAD plus to NADH. By converting this pyruvate to acetyl-CoA, we can enter the Krebs cycle, and by producing this CO2, we've done the first respiration step. So the process of releasing the CO2 is part of respiration. Then we get into the TCA cycle or Krebs cycle. They are the same thing, they just have different names. And this is showing you that pyruvate, and then this is the conversion to acetyl-CoA, which then gets put in here to citrate, and we go all the way through this process. All right. As you go through this process, you'll see there are some important things. We get an NAD plus to NADH here. We get another one here. We get one production of GTP here. GTP functions like ATP. And then we get NAD plus to NADH here. And this is going to happen 2 times. All right. So we're going to, and the reason it happens 2 times is that you get 2 pyruvates for every glucose, right? So each of those can go through this process, can fuel this process 1 full time. And so you may hear this called TCA cycle, Krebs cycle, citric acid cycle. They're all the same thing. And so this is what I was telling you about the need for acetyl-CoA. By doing this, you're going to ultimately get 1 ATP or GTP, right? You get it here. And then you're going to get the 3 NADHs, 1, 2, 3. And then you're going to get 1 FADH2, which is going to be represented here, I believe. And then it also produces 2 CO2 molecules, right? We've got a CO2 here and a CO2 here. It's going to do that for each of your pyruvates, so that gets doubled. All right. And so now that we finished the Krebs cycle, and let me go back here. I do not expect you to know every one of these steps and every one of these enzymes in the Krebs cycle or in glycolysis. I want you to have a general understanding of how this process is working, okay? And what is it doing? So I need you to know what you're producing here, and I need you to know that acetyl-CoA is necessary for these processes to be carried out in the Krebs cycle, and you get that acetyl-CoA from converting pyruvate to acetyl-CoA in the intermediate step. And so here, we've got these different respiratory chains. They're going to have a set of coordinated redox reactions that are basically going to exchange electrons down a chain, thus the electron transfer chain. And this is going to be what is used in oxidative phosphorylation and photophosphorylation. I'm not going to talk too much about photophosphorylation, primarily oxidative phosphorylation. All right, so here you go. You've got your electron transport chain. You've got these NADHs, right? So these are reduced molecules. They gained their electrons, right? And they're going to feed into this chain of proteins. And as they feed in, they're going to pump hydrogen ions, protons, out of the cell, all right? And they're going to get recharged to their previous form, so back to NAD+. Or FADH2, which enters here, is going to become FAD, and that is going to also go back into the Krebs cycle. And as they go through, as the electrons are transferred along this red line, they ultimately are going to end on your terminal electron acceptor, in this case, oxygen. It's going to result in this proton gradient outside of the cell. And then as the protons pump back into the cell through ATP synthase, ADP is going to be phosphorylated to ATP. And so this is a basic explanation of that final electron acceptor. Aerobic respiration chains use aerobic cellular respiration, and therefore they need oxygen as a final electron acceptor. Anaerobic respiratory chains do not use oxygen, so they need something else. It could be nitrite, nitrate, it could be some sort of iron, etc. Now, for any of this to happen, you have to have these membrane-associated electron carriers. Those are all those proteins that are transferring the electrons along the cell membrane. And so this is again showing you that NADH, the electrons are going to come in here, transfer through here, out here, onto oxygen. I don't need you to know all of these enzymes. I don't need you to know cytochrome C oxidase. I just need you to generally understand that the electrons are transferred through these membrane-bound proteins, and as they do so, the hydrogen ions are being pumped out of the cell. That creates a proton gradient that can then be pumped back through ATP synthase, phosphorylating ADP to ATP. And also understanding that the oxygen is your best electron acceptor, and you get more you get more protons pumped out of the cell with the better your electron acceptor is. And therefore, oxygen is the best electron acceptor out of all of our available electron acceptors. It's going to produce the most ATP. Anaerobic respiration is going to produce less ATP because it doesn't have as good of an electron acceptor. And here is an example of your different electron acceptors. So the lower down this list, the better the electron acceptor is. And so the process goes this way: O2 to H2O, Fe3 plus can become Fe2 plus, nitrate can become nitrite, fumarate can become succinate, sulfate can become hydrogen sulfide, CO2 can become CH4, sulfur can be hydrogen sulfide, and then CO2 can also become some sort of carboxylic acid. All right, so what's going to happen is that if your terminal electron acceptor is here, you're going to produce more ATP than if you had iron. If it's iron, it's going to be more than if you had nitrate. And so again, it's all dependent on what kind of organism it is, and some of them can actually have multiple electron acceptors depending on whether or not or what kind of environment they're in. And so this is just reiterating that you generate that proton gradient, and this is forming what's known as the proton motive force. As those protons are pumped back in, they generate ATP. That process is called chemiosmosis. This is the flow of protons back into the cell allowing ATP to be phosphorylated allowing ADP to be phosphorylated to ATP. All right. And so here's like a close up. It shows that this is in the ATP synthase. This is actually like a rotor, and it gets spun around as these hydrogen ions get pumped into the cell. And that spinning is what generates the energy to create this bond here. All right. And so chemiosmosis is this important process of making ATP. That is how you're going to get ATP during oxidative phosphorylation and photophosphorylation. And this is basically just kind of summarizing what I showed you in those images, that the reduced NADH and FADH2 drop their electrons off in that chain. And then they go through a series of redox reactions that are going to keep transferring those protons. And they move from this high energy state to low energy state, ultimately ending up on the terminal electron acceptor and pumping those hydrogen ions out that can then be pumped back in to fuel ATP synthase. Here's a summary of what you get out of aerobic respiration. You'll see that we get 2 ATP invested at glycolysis. Between all of the cycles, we produce 40 ATP, or up to 40, and then that gives us a net of 38, 40 minus 2. And then you can summarize the overall reaction like this, right? I don't expect you to know that, but it's just kind of fun to look at how what you actually need to do cellular respiration. We need our 1 glucose molecule, we need our 6 diatomic oxygens, 38 ADP with 38 phosphates, which will result in our 6 CO2 and our 6 water molecules, as well as 38 ATP. So we get 1 water molecule per carbon in our glucose. Or sorry, 1 CO2 and water molecule for each of them. And this is to reiterate where they are. In eukaryotic cells, the electron transport chain is going to be in the membrane of the mitochondria. In prokaryotes, it's going to be in the plasma membrane. And we can have multiple types of electron transport chains in individual microbes. This is one way that we can actually identify organisms, is what kind of electron transport chains they have. We have a test called the oxidase test, and it's actually looking for the presence of cytochrome C oxidase. Here's another way to look at that. Keep in mind that I don't necessarily need you to memorize all this stuff. I'm just kind of showing you how it goes together. And you should, at this point, be able to meet all of these objectives. I'd like you to write down some of the questions you have about stuff that isn't clear to you at the end of this video, and bring those to class so we can talk about them and hopefully bring some clarity. All right, I hope you enjoyed, and I'll talk to you soon.